Today, we will present our science communication poster based on the article Effect of Different Bulking Agents on Feed Batch Composting and Microbial Community Profile. I will start with the introduction. So food waste has been a major environmental concern in China due to its improper disposal method like landfilling and incineration that has caused pollution and loss of resources. Since food waste has a high organic content and nutrient, so it is valuable resource if recycled effectively through decomposing. Based on the research, feed batch composting is a sustainable on-site treatment in a small reactor. So bulking agent has been used to enhance composting efficiency and the quality of the product. So in the article, 4 types of bulking agent with different size has been used, which are green waste, wheat straw, sawdust, and bamboo charcoal. So in the previous research that has been examined, the effect of the bulking agent type, particle size, and reactor temperature on feed batch composting has been remained unclear. So in this research is to aim to compare the performance of different type of bulking agents. Secondly is to compare the effect of different size bulking agent on feed batch composting properties. And lastly is to investigate the influence of temperature on microbial communities during the composting. So the next methodology, I will pass it to Sebastian Tua.
So in the methodology, the first step is composting materials and fed batch reactor. The food waste and the green waste were being collected, and both of them are put in the reactor. Secondly, experimental design and sampling, which is A1 represent green waste, A2 wheat straw, A3 sawdust, and A4 is bamboo charcoal. Consists of 3 experiments. The bulking agent were only fed into the reactors on first day of composting. The third step is analysis of 50 chemical properties. The moisture was determined by drying the samples in an oven at 105 degrees Celsius for entire night. The moisture was then calculated. The fourth step is the microbial community analysis, divided into 2 sections, which is section A, 805 R primers and 341 F primers used to amplify bacteria in sequencing platform. Section B, primers compatible with Illumina Bridge PCR were utilized for amplification. Last but not least is the one-way analysis of variance, ANOVA, with features least significant difference are used in the last step, which is statistical analysis. Now we are to the next person with result and discussion.
So for the result and discussion, in the result, they stated that they used 3 bulking agents, which were sawdust, rich husk, and cornstalk, to see how they affected food waste composting in a fed batch system. We found that sawdust performed the best, achieving the highest temperature peak and maintaining thermophilic condition the longest. This led to more efficient organic matter degradation, shown in the compost lower final total organic carbon and higher germination index. In contrast, cornstalk resulted in the highest bacterial diversity based on Shannon and Simpson index. While it didn't compost as efficiently as sawdust. It supported a broader microbial community, which may benefit soil biodiversity. In the discussion, we point out that these differences are largely due to physical properties. Sawdust's fine texture help regulate moisture and aeration, supporting microbial activity and heat retention. Cornstalk, being more porous and fibrous, likely created microhabitats that favor microbial variety. So overall, sawdust is ideal for faster composting and better maturity, while cornstalk promotes microbial richness. Depending on your composing goals, whether it's speed or biodiversity, different bulking agents offer different strengths. That's all. Thank you.
So for the conclusion, the article research that we have found is it has been achieved the three main objectives that has been discussed in the introduction, which are the first one is green waste is the one work optimally as a bulking agent on feed batch composting due to its optimal moisture content, weight loss rate, and CN ratio. Secondly, the 3 until 6 millimeter green waste has helped to lower the moisture in the compost and enhance the organic matter decomposition. Thirdly, which is the last, it is increasing in reactor temperature are conductive to thermophilic bacteria and fungi, which it has contributed to cellulose and lignocellulose degradation. So lastly, School of Civil Engineering and Southeast University, Nanjing, China. And secondly is the IHE Dell Institute for Water Education has been supported throughout the research. Thank you. That's all from our group.